Hi, Denise Caldwell here from Effectual Services. Glad you've joined me for this IP Minute to Go. On this special extended edition, we're going to take a few extra minutes to cover some technical subject matter. Our topic today is sequence searching, and the content has been developed by Rohit Agarwal and our Life Sciences team at Effectual Services. As you probably know, DNA contains all the information needed to make the proteins, and RNA acts as a messenger that carries this information. The conversion of DNA to RNA is known as transcription, and they are further translated to proteins. A sequence can be of two types, a nucleotide or a peptide sequence. A nucleotide sequence is a series of alphabetical letters that indicate the order of nucleotides within a DNA or RNA molecule, where nucleotides are the building blocks of nucleic acids. A peptide sequence, also known as an amino acid sequence, is the order in which amino acid residues are connected by peptide bonds. Shown here graphically, you will see a nucleotide sequence can be a primer, probe, or biomarker. A primer is a short nucleic acid sequence that provides a commencement for DNA synthesis. It's used to synthesize double-stranded DNA from single-stranded RNA using PCR. The length of primers is not usually more than 30 nucleotides. A probe is a fragment of DNA or RNA of any length varying between 100 and 1,000 bases. It can be radioactively labeled and is used to detect DNA which is complementary to the sequence of the probe. A peptide sequence can be an antibody sequence or a T-cell receptor sequence. An antibody sequence is an amino acid chain of varying length between 100 and 1,000 bases that binds to an antigen, toxin, or foreign substance to produce an immune response in the body of a living organism. While filing patent applications containing nucleotide or peptide sequences, you must list these sequences separately and in a proper format such as FASTA. FASTA is a text-based format representing nucleotide or peptide sequences. These sequences are represented using single alphabetical letters. There are certain rules and regulations laid out by each local patent office for sequence listings of nucleotides or amino acids. The sequence listing shall be referred to by the sequence identifier, that is a unique integer that corresponds to the sequence ID number assigned to each sequence in the listing. If furnished on paper, it must be independently page numbered, and if furnished electronically, it must of course be in proper format and accompanied by an appropriate transmittal. A nucleotide sequence shall be presented only by a single strand in the 5' end to 3' end direction from left to right. The terms 3' and 5' must not be represented in the sequence. Please take a look at the arrow in the lower center part of the slide for an example of some of the mandatory fields that are required by various local patent offices. These can include applicant name, title, the number of sequence ID numbers, length, organism, and so forth. To standardize sequence presentation, patent offices have developed and offer various software programs which are publicly available, such as BISAP or Patent-In, developed by the EPO and the USPTO respectively. So why are sequence patents so important? There are more than 40,000 patents on DNA molecules. Nearly 20% of the human genome of the known 23,000 plus human genes have been patented, and out of these, half of them are owned by private companies, represented by industries such as healthcare, pharmaceutical, diagnostics, and biotechnology companies. Sequence searching is required generally in projects related to patentability searches, product clearance, infringement, and invalidation searches. To properly perform searches for patents or NPL with sequences, a sequence alignment exercise is a must. Sequence alignment is an algorithm such as BLAST to establish similarity between two sequences, or put more simply, it is a character-to-character matching exercise. You'll see there are certain challenges faced by the industry while conducting sequence-based searches, cost being a major factor, lack of expertise within the domain. Sequences present in the patents are in non-editable format, and there are only limited databases to identify sequences disclosed in patents. Generally, there's no uniformity in submission requirements. We can search NPL containing sequences, but access to full text is not available. It can be difficult to access patented sequences, especially in foreign jurisdictions, and there are very few sequence alignment tools. You'll see here there are free as well as paid databases to perform sequence searches. Examples of freely available databases are NCBI BLAST and Patent Lens. BLAST represents Basic Local Alignment Search Tool and is an algorithm for comparing primary biological sequence information such as amino acid sequences of proteins or the nucleotides of DNA sequences. Patent Lens allows you to search over 80 million DNA and protein sequences disclosed in patents. Examples of paid databases are GenomeQuest or those hosted by STN, such as DGene, USGene, PCTGene, and the like. With the STN group, sequences can be searched in patent and non-patent literature, and it provides access to a broad range of databases. With GenomeQuest, you are limited to patents only with no NPL capability. I hope you have found this to be a helpful IP Minute to Go. We're so glad you've joined us.